Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stephan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Stephan Hamperl from the Helmholtz Zentrum in Munich on this show. Please let me briefly introduce you to our audience. You did your undergrad studies at the University of Regensburg, where you also performed your PhD research with Dr. Joachim Griesenbeck. You then moved to Stanford University in California to conduct your postdoc in Karlene Simprick's lab before returning to Germany to establish your own group at the Institute of Epigenetics and Stem Cells at the Helmholtz Zentrum in Munich in March 2018. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
Yeah, that's a very interesting question. I think I got really inspired the very first time during high school. And I was in the chemistry class, and I was really inspired by our chemistry teacher. So he was very, very passionate about distributing the different topics and delivering the topics to us. And I got really inspired and got interested into chemistry, and that actually allowed me, or kind of let me decide in the beginning to study chemistry and not biology.
And when did you then, did you study chemistry until the end, or was it just biology? Or did you switch to biology? That's the question.
I did switch, actually. So after my high school degree, I had to do my military service for 9 months. But then after that, I signed up for chemistry at the University of Würzburg for 1 semester. And I was kind of happy with this. But then at some point, I saw that the University of Regensburg had this biochemistry class that seemed to be a very interesting topic. And I started to get interested in this as well. And that's why I applied. And at the end, I switched after 1 semester from chemistry to biochemistry.
Yeah, I guess that's what most people do, right? Or that end up in epigenetics, that they studied kind of biochemistry or molecular biology, something around that.
Right. Yeah.
So let's come to your science that centers around how DNA replication and transcription can occur simultaneously without major accidents that cause DNA damage and genome instability. Maybe we can start in the year 2017. There you reported an isolation protocol for the targeted purification of specific genomic regions in their native chromatin context from the yeast Saccharomyces cerevisiae. I guess that was like maybe the first work out of your PhD. And can you maybe talk about the study and how you then transitioned or how you got interested in exactly this topic?
Absolutely. So I kind of started working on this during my diploma thesis. I was finishing my biochemistry class at the University of Regensburg, and then in my diploma, which would be consistent with a master thesis, I got interested in this research topic, and particularly what fascinated me was to this method that we developed or started to develop to specifically recombine a chromosomal domain from the yeast genome by site-specific recombination. So we would create a yeast strain that contains recombination sites, and upon expression of a recombinase, we can specifically target a domain and isolate it, and then define what kind of proteins are associated with this particular domain. So kind of like a reverse, what people know as a chip experiment, where you look at a specific modification or factor and you're interested in where in the genome it binds. I found what fascinated me at the time is that you do the opposite. Instead of checking all the binding sites of a factor, just looking at a particular region and finding out in one experiment all the target proteins and histone modifications or whatever you're interested in, and finding them all in one single experiment. And this was the fascinating thing for me that I really wanted to pursue. And we took us quite some time during my PhD to really develop the method, but eventually we got there to a level where we could say, okay, this is indeed the locus specific proteome of that region that we wanted to target at the time.
Were you able to make certain discoveries using that already, or was it just focusing on the method development?
We could identify a few new factors that haven't been described at the time, binding to this locus. And in particular, there were some chromatin remodeling machines. One of them, the Ino-80 chromatin remodeler, that hasn't been described to be specifically localized at this site. And so this is something that we identified newly in the data. And so it was both method development, but also we could really identify a bit of a new biology with the system.
Next, and this was the first paper out of your postdoc, you switched gears a little bit. Maybe you can also, I mean, it's always interesting, especially for younger people, to get some information how people what their thoughts were in the transition from the PhD to the postdoc. So maybe you can talk about the transition and what you got into then.
Absolutely. So I would say my PhD thesis was very biochemistry heavy and very targeted in that sense. We really wanted to get this method established. So in a way, it was really hardcore biochemistry on yeast as a model organism. And I think I would say there are two major motivations for me to switch to this postdoc or to this project in the postdoc. For one, I really wanted to go away from yeast as a model organism and experience a bit also higher eukaryotic genomes, mammalian cell culture. And the second motivation for me was the topic itself, because I thought it intriguing that DNA replication and genomic instability that they are so intertwined, but we really still don't have a very good understanding of it. So it was also a complete switch of scientific fields for me. And also in terms of methods, I really switched from biochemistry to a lot of cell biology methods. So it also increased a bit my repertoire of methods and techniques that I could use.
Yeah, when we go more into the details of what you did, you investigated how conflicts between transcription and replication, as I already mentioned too, and R-loop formation influence genome stability in human cells. And at least at Active Motif and maybe also in the field of epigenetics, R-loops are kind of getting more attention in the last couple of months or years. Maybe you could give us a short introduction into R-loops and tell us what you've then found investigating this.
Mm-hmm. Yeah. So R-loops are considered as a type of a secondary DNA structure. So they can occur at actively transcribed genes, and they can form when the nascent RNA that is just protruding out from the RNA polymerase, that it reinvades in the DNA duplex behind the RNA polymerase to the complementary non-template strand. So in this way, it creates like a three-stranded nucleic acid structure composed of this RNA-DNA hybrid plus a tract of single-stranded DNA around it. And this is what we call an R-loop structure. And in the past, like let's say 10, 15 years ago, R-loops have been described already, but they were more like considered as like kind of a transcriptional byproduct, which is, it happens sometimes, it quickly gets dissolved, and there were not really associated any major consequences or functional roles to this. But this view has really changed over the last, I would say, 10 years, where it was shown in many labs across many publications now that under conditions where you have an increased level of these R-loop structures, these cells have also very, very strong signs of genomic instability. And that was really the interesting question to ask when I started my postdoc, because just the presence of the secondary structure doesn't really mean, so why should there damage arise from these particular structures? We didn't really know or understand how then DNA damage and double-strand breaks are occurring just because we have an elevated level of these structures. And the hypothesis we pursued is maybe it is when these cells replicate and R loops are in the way of an approaching DNA replication fork that this interferes and blocks replication fork progression, and that could lead to DNA damage. And this is exactly what we wanted to test in my postdoc and show.
Can you talk about the results then? Because I guess that's very interesting, because it was now, I guess it was, and this is a modification of my next question, but so this was then basically the modification of just an observation to getting to a physiological relevance, right, of the R loop structure.
Yeah, so what we decided to do and what we found out with the system is, so one issue in this field is that we have no good way to really control DNA replication and transcription to really collide at the same locus at the same time. It's DNA replication on its own is.
Sorry, why would they collide? I mean, they are not coming from the different directions, right? They are. I mean, that there must be some sterical issues, and I mean, RNA polymerase and the replicative enzyme. I mean, there must be enough space for both enzymes to be there, right?
So that's actually a good question, and so I think one issue here is that you have many across our genomes. You have many sites of DNA replication initiation, but also many genes that are actively transcribed. So per se, these two machineries are very pervasive. And so from this, therefore, they can also meet in different orientations. And it's also probably stochastic when and where they meet, depending on the current replication and transcription status in the cell. So that's one complexity that is really difficult to address. And the kind of solution we did is to simplify the whole system and focus on an episomal system where we cloned one transcription unit next to one replication origin and really then looked what happens if a replication fork approaches this particular transcription unit in either a head-on orientation or co-direction orientation. And that was the major system that we used in my postdoc.
So before we go to the results of that, so the R loop can only be formed by RNA polymerase, right? Because the R loop is formed by definition by 2 DNA strands and 1 RNA strand then.
Exactly. Yeah. That's the current model, yeah.
And what did you find then using the system?
So what we found is that it really matters how the replication fork approaches an R loop, in which orientation. So if these two machineries collide in head-on orientation, then the two machineries are blocked and they are causing persistent blockage of a replication fork, and that causes a major DNA damage response and DNA breaks. But in the co-direction orientation, so when the replication fork approaches the R loop in the same direction, then the replisome itself is actually able to remove the RNA DNA hybrid and kind of relieve the R loop levels. So it seems that the replisome has an intrinsic RNA DNA hybrid resolution function that hasn't been discovered before, but it really depends on the orientation. This can only occur in the co-direction orientation and not in head-on. And that was one of the major findings in my postdoc.
So you're saying that the two machineries collide, but the R loop, so the R loop itself, is it only like the nucleic acid structure, or are there proteins attached to the R loop?
At the time, so we mostly focused really on the physical changes to the DNA. Are there any breaks? Are there, what happens to the DNA? And we didn't really consider any other proteins associated, what happens to the local chromatin structure. So this is something we started to investigate only later when I also started my own lab. But at the time during my postdoc, we mostly focused really on the physical impact of these conflicts onto the DNA.
So then I guess you moved to Munich. And I guess it was there where you developed the assay, which was called MATAC-Seq, which stands for Methylation Accessibility of Targeted Chromatin Domain Sequencing. Can you give us a walkthrough of this method and how you got interested in that? Because I guess that's a little bit different than what you did with the R loops.
Correct, yeah. And maybe I also should kind of give a short recap when I started my lab. So when I got this position here at Helmholtz Munich, I really wanted to combine the expertise of both my time doing my PhD, but also during my postdoc. Because as I mentioned before, we had these very distinct projects, but also distinct methods and techniques. But I felt in my own lab, I would really like to combine the expertise from both of these systems. And what I really liked about my PhD project is that this method had really, in my opinion, there was a lot of power in this method because you could target any domain or region of interest that you would like and understanding what kind of chromatin factors are associated with this particular region that you're interested in. During my postdoc, I kind of stopped working on this and I really wanted to get back to this. And what you mentioned, Stephan, about the MATAC-Seq, this was actually a project where we actually went back to this yeast chromatin purification project that we simply applied to another specific locus in the genome, namely replication origins. And what we did in this study is that we purified distinct chromatin domains. In this case, we targeted distinct DNA replication origins, so the initiation site of DNA replication. And in particular, we targeted 4 distinct sites. 2 of the origins are known to be early and efficiently replicating, and the other 2 are late and inefficiently replicating. And it's the big mystery in the DNA replication field to understand why a certain replication origin would fire early in S phase and the other would fire late in S phase. And we thought, okay, maybe it's the chromatin structure associated with these distinct origins that would define this replication time. But how would we test this? So one idea was to purify these domains and then check the chromatin accessibility of these sites. But of course, this has been done before. And MATAC-Seq, the interesting point about, so this is a nanopore sequencing based readout where we first isolate these chromatin domains and then we incubate them with a cocktail of methyltransferoses that will methylate the linker DNA between nucleosomes, but not for nucleosomal DNA. And with the readout of this adenine methylation, we can then pinpoint or track back where on these single reads that we get was the nucleosomal imprint and the linker DNA imprint based on the DNA methylation levels that we can read out. And the cool thing about this method is that it's no longer like a bulk sequencing method, but it is actually a single molecule readout. So we get for each single molecule read that we have, we get a clear, distinct profile where on this read the nucleosomes were sitting. And in this way, you can also reveal the potential heterogeneity of molecules where the nucleus are exactly positioned. And this is exactly what we found with these replication origins, that even though we isolated these domains in a very specific cell cycle stage, we expected to be very homogeneous, but we actually identified like a large heterogeneity, that many of these molecules had very distinct nucleosomal profiles from like very nucleosomal to very accessible. And we think that this kind of heterogeneity that we observed could be a link to why a replication initiation is to some extent considered a stochastic process, because you have this large heterogeneity in this cohort of molecules. So this is one main finding of this study.
And I guess you You're still working on that, right? And developing this further and using that method for future projects.
Exactly. Yeah, we are actively working with this method, and kind of the next step that we have one ongoing project now in the lab is so we collaborate with a lab at the DMC, Christoph Kurat's lab, where they developed an in vitro replication system, and we are trying to, after isolating these native domains with our approach, we try to in vitro replicate this domain and see whether, for example, an early or a late origin has more efficient replication. And these are kind of follow-up experiments that we're doing in the lab.
Yeah. So if the process is stochastic, then the cell must have some kind of measure to measure which sites have already been replicated, right? I mean, if you have a clear plan, then you know what you're doing. But if you don't have a clear plan, then you probably don't know what you're doing. And how does the cell know which ones still need to be replicated?
So that's actually kind of solved by the cell itself, because there is a clear distinction between the origin licensing and the origin firing, and that's mediated by the cell cycle. So origin licensing can only take place in G1 phase of the cell cycle, and origin firing can only take place in the S phase of the cell cycle. And if you would want to re-replicate something, you would have to relicense an origin in S phase, which is suppressed by certain kinases that are activated and specifically phosphorylate. So in a way, the cell has developed a very nice backup system to exactly prevent this.
So next, and this paper was published early this year, so early 2024. You described, again, another method and maybe an orthogonal method to what you have been doing, an automated image analysis pipeline to quantify the coordination and overlap of transcription and replication activity in mammalian genomes. So, I mean, microscopes are very expensive. so how did you get into that, and how does this work?
Right. So I think this paper is then based on kind of what I said about also taking the experience that I had during my postdoc and continue working on this transcription replication conflicts. And when I started here in Munich, one of the issues we faced is that so far, really having good methods in hand to visualize active transcription and replication simultaneously in a cell is certainly was at least a bottleneck that we thought we want to address or try to solve. And what we developed is a very, I think for the community, very useful tool where you have an automated script that you can download and then simply quantify the level of signal that you get in one channel in a microscopy experiment versus the other. And you have many parameters that you can play around and get quantitative data out of this. And we particularly use this to analyze the overlap of transcription and replication using specific stainings in a nucleus.
Which stainings would you use? And did you also stain for R loops?
Yes, we also did quite some R loop stainings. But for this kind of sites of active transcription replication, for replication, we use a short pulse of EDU that we can then click on a fluorophore. And for active transcription, we use actually an antibody against an elongating version of the RNA polymerase 2, so phosphoserine 2, modified RNA P2.
And for the R loops, you use the S6.9?
Yeah. We use this S10.6 antibody, right?
No. And it worked well in your hands?
So there have been a bit of discrepancies in the field. Maybe you're aware of this, Stephan, that this about the specificity of the antibody. So it does recognize to some extent double-stranded RNA. And I think especially for immunofluorescence staining, it is very important to do these stainings in the right conditions. So one of the key things we always do in stainings with S10.6 is the RNase A digestion. to remove potential RNAs that can increase the background of the staining. And we have to do this in a very high concentration of salt, 500 millimolar sodium chloride, because if you do it at a lower salt concentration, the RNase A has also RNase H-like activity, so it can also degrade your RNA-DNA hybrids. So that's, of course, an issue. And this is something that needs to be carefully controlled in this case.
Okay. So imagine you are due to submitting a grant proposal tomorrow, and maybe you are indeed. What would you have written into that?
But I see what I would like to continue to research in the next 5-10 years is definitely the transcription replication conflicts. We have now collected some really good evidence that locally, when you look at the conflict site, that this really impacts the local chromatin structure. These sites are depleted or become depleted of nucleosomes. They're very accessible and therefore probably also more prone to DNA damage. And we are looking at what kind of epigenetic modifiers are potentially recruited at these sites. And this is one line of research we want to investigate further to really get a molecular understanding what a TSC site really is and what kind of changes of the chromatin are occurring there. And the other kind of second line that I'm really interested in, so far, many of the studies are mostly focusing on certain cancer cell lines that are already kind of messed up in their genome and potentially have already more TSCs to start with, let's say. So one interest of one kind of follow-up we would really like to do is actually to go into what are considered more normal cell lines with a normal karyotype, so non-cancerous cells, and ideally identify to what extent if we provoke transcription replication conflicts, for example, by activation of certain oncogenes, that's one example, whether that could drive, that TSCs could be driver events to transform these cells into a cancer cell-like cell. And this would be one kind of idea that we would love to pursue in the next couple of years to come.
So your research is more like not in those stages where you would have like a treatment for a disease as a goal, but rather still in the early stages where it's still like fundamental research. Could we see?
It's definitely, I would see this as what we do is fundamental research. So at this point, we don't have any kind of translational applications in the lab. Although I should say that there has been a recent study on a PCNA inhibitor. So PCNA is a component of a replisome. And this particular PCNA inhibitor is shown to increase transcription replication conflicts specifically. So there could be, in the long term, I think, some interesting applications that the TSCs may play a role also in human diseases. But really, our focus in the lab is really to understand the molecular basics and how, for example, these TSC sites are then resolved, the pathways are activated. But if we have this information in hand, I do think that this could be a relevant information also in the future to develop certain clinical applications to target such pathways that would be important for this. So I do see in a very, very long perspective also some potential here.
Now, when you're talking about those conflicts, is this, I mean, we have three players, right? We have the replisome, we have the RNA polymerase, and we have the R loops. So is this always a combination of all those three, or are you specifically looking at only the enzymes that collide?
Yeah, so what we think is actually there's also the potential that there are conflicts without R loops, so only the two players involved. But I think these are much less deleterious than the ones that involve an R loop. And the reason for this is that the R loop itself is a very stable structure. And the replisome, at least in the head-on orientation, it can actually stabilize the structure even more. We don't know exactly the mechanistic basis for this, but we see at conflict sites a clear association of more RNA-DNA hybrids. Whether this is cause or consequence needs to be debated, that we don't know. But there is certainly a correlation that conflict sites that are highly prone to DNA damage and replication stress, they are more associated with R loops. But that does not mean that conflicts may not also occur without the RNA-DNA hybrids that are formed. But what we've shown in the postdoc study, so we also had one sequence that is not prone to R loop formation that we cloned in these vectors. And there we didn't see any DNA damage response. So suggesting that really the R loop is a very strong contributing factor to the DNA damage of conflicts.
Yeah, I mean, the R loop is a static structure, right? And the enzymes are very dynamic because they have to move along the DNA. So there might be something going on differently with the R loop and the enzymes themselves.
Exactly, yeah.
So for the last 30 minutes, we have been on a journey through your scientific career. Did you miss something important, or would you like to add something? I hear you thinking.
I think what I can say is that I was really lucky for the different positions and throughout my academic journey so far. And I was particularly lucky with my mentors, supervisors, both in my PhD and postdoc. And I think the key for me is that they were not only supervisors, but they were really mentors that I'm still in touch now, and they continue to support me. And I think this was one of the key points for the success we had so far. So and it's maybe more on a personal note, I guess.
Yeah, that's good. That's good. To finish off, could you give us a brief summary of what you would consider your most important finding?
So I think one of our key contributions is that indeed the replisome, if it detects or if it encounters an RNA-DNA hybrid containing RNA-P complex, that is a bad thing for the cell, in particular when they face each other in a head-on orientation. But then it can also be a good thing because in the co-directional mutation, it can help to remove these structures, which is something not really predicted. This, I think, can explain also why majority of mammalian genomes are set up in a way that there is a bias towards co-directionality of replication and transcription. And to do exactly this, that to help transcription and replication to be well coordinated in the genome and not interfere too much by promoting more of these co-directional conflicts. And this is one of our major contributions. And the second is that this definitely also affects the underlying chromatin. So it not only affects the DNA, it also affects the chromatin. And this is something that hasn't been looked into so far, but there are a lot of changes, many dynamics happening at these sites that we still don't really understand. And this will keep us busy to figure this out in more detail over the next couple of years, I think.
And that's also a good thing. So,
exactly.
Thank you, Stephan, for your time and for being on the show.
Thank you very much, Stephan.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.